So for people who are just walking in, and thank you for coming here today and to the meeting. As I was saying earlier, just to recap, the theme of this meeting is, tripartite. And today's theme really focuses on positive emotions and the multimodal tools and techniques that are being used to kind of unravel and elucidate the neural mechanisms underlying behaviors associated with positive emotion and reward. And it's my pleasure now to introduce the first plenary speaker of today and of this first plenary session. This is Garret Stuber, and let me just tell you give you a few words about Garret. Garret's professor at the University of Washington and at the University of Washington Center for Neurobiology of Addiction, Pain and Emotion. He's had a really stellar and amazingly rapid rise in fame over the last few years, such that really he's now one of the leading stars in his area. He started his own lab only as recently as 2010 in cell biology and physiology at the University of North Carolina in Chapel Hill before moving to the University of Washington last year. Now, he's already been the recipient of a very prestigious NIDA Merit Award and also the very prestigious Woletsky Award from the Society of Neuroscience in 2017. Garret's lab really focuses on using multimodal, cutting-edge techniques, both in vivo and in vitro, to really hone down and elucidate in fine detail the neural circuit mechanisms that underlie adaptive and maladaptive behavior states. So it's my pleasure today to introduce Garret, who's going to come and tell us about dissecting the neural circuits for motivated behavior. So Garret Stuber.
All right, thank you very much, Mary, for that wonderful introduction. It's a real pleasure to be here and to share some of the work that we've done over the last few years in my lab. It's also a real big pleasure to follow Eric Nestler's talk. I've always found his work since the beginning of my career to be really inspiring and visionary for the field. And I think what I'll have to say will pair nicely with some of the research that he highlighted. So my lab's interested in the neural circuitry that controls various types of motivated behavior. To conduct this work, we're a basic neuroscience lab that uses animal models to investigate reward-related behaviors and different types of motivated states. Thinking a little bit about motivated behavior, there's been a number of theories over the years in terms of how we should really conceptualize this. For example, the psychologist Abraham Maslow posited that there's a hierarchy of needs that every organism has, starting at the very basic foundational physiological and safety needs of an organism that must be met first prior to building up to more human-specific motivations, such as creativity and other aspects. The physiologist Clark Hull put forward the drive reduction theory of motivation, which basically states that an organism is motivated to perform a particular behavior as some sort of signal, a biological signal that conveys drive for a particular type of motivation. As that rises, this facilitates the drive to perform that behavior. And then finally, the psychologist B.F. Skinner, as we know, put forward the idea of positive and negative reinforcement, and that, and this is really important because I think it conveys the fact that valence is really critical for different types of motivated behaviors. So not only do we have positive reinforcement or appetitive behaviors such as reward seeking, but it's also important to consider aversive behavioral states as well, such as escape and fear. In my lab, we study the biological basis of motivation, and specifically we're focused on trying to understand the precise neural circuit architecture that can control different aspects of these types of behaviors. And so what I mean by this is really trying to identify the genetically specified cell types within the brain that are critical for different types of behavior. In addition to just trying to elucidate these cells, it's also important to consider how these cells in the awake and behaving animal are encoding different aspects of motivation. How do they respond to motivationally relevant stimuli? How are they modulated by the biological state of the animal? And so to do this, we've used really cutting-edge tools within systems neuroscience to begin to elucidate these functions. Now, this is critical because not only is motivation really critical for all aspects of life, but we know that it's also highly dysregulated in many forms of mental illness and other disease states. And so we think that by understanding the circuit function and how it's altered in different preclinical animal models will give us hints and a better understanding of how neural circuit function is relating to neuropsychiatric illness. Okay, so when we start thinking about the biological substrates of motivation, what brain regions in particular are we talking about? Most of our work has focused on a critical network of subcortical brain regions that are known to be highly essential for driving both positive and negative motivated behavioral states. These brain regions, probably many of you are familiar with, include the basal forebrain, the ventral midbrain, the hypothalamus, and many other key subcortical sites that have been shown over many studies over many decades now to be critical for driving different types of motivated behavior. While these areas are essential for this, we also know that they don't function in isolation. And in fact, they require input from virtually all other parts of the brain. In particular, sensory and higher order cortical areas either directly or indirectly impact these motivation control centers to modulate their activity and presumably to convey important sensory information as well as just the cognitive information that would be critical and is needed to tap into the motivational reward circuits in the brain. Additionally, we know that the activity within motivation centers can be modulated and impacted by circulating factors from the rest of the body. So while the brain is critical for driving this, it's important to consider that signaling molecules, mostly endocrine molecules, hormones, and cytokines, are also very powerful at impacting the activity of these neural circuits. Ultimately, we think that these signals are processed in these subcortical areas and then conveyed through the basal ganglia, brainstem, and spinal output circuits to control observable actions. Okay, so with that in mind, what I'd like to do today is to highlight some of the work that we've done over the last 9 years or so that's really started to begin to elucidate some of the critical neural circuits that are important for driving motivated behaviors. So I'll just very briefly start by mentioning the midbrain dopamine system, which we all know is critical for reward seeking, but also plays an important role in other types of behavioral states as well. From the dopamine system, what I'll do is work back from those cells and discuss critical regulators of the dopamine system. In particular, I'll talk about our work on the ventral tegmental area Gabaergic neurons and how we think that this is an important negative regulator of dopamine neuron activity. After that, I'll talk about another critical input to the ventral tegmental area, the lateral habenula. This is a brain structure that's known to be highly involved in aversive behavioral states, And I'll mention some of our work on the circuit mechanisms on how we think this is performing this. And then finally, I'll end with two other inputs to the VTA that we think are critical, or inputs within these circuits that we think are critical. Those from the extended amygdala, or the bed nucleus of the stria terminalis, as well as inputs from the lateral hypothalamic area. Okay, so I think it's universally agreed upon that dopamine release from midbrain dopamine neurons, in particular those from the ventral tegmental area, serves as a critical and potent signal for controlling learning and motivation. We know from really decades of work from many pharmacological and genetic and lesion studies that manipulating the dopamine system really produces profound effects on reward-seeking behavior, as well as effort and vigor for responding for particular rewards. Now, while it's established that the dopamine system is really critical for driving reward and motivation, what's less clear is what's tapping into the dopamine system. So there's been some really beautiful anatomy studies over the years, and I'm just highlighting one in this image from the Uchida lab at Harvard, which has shown that there's a variety of subcortical brain regions that are either directly or indirectly interfacing with the midbrain dopamine system. So in this image, you can see that cells that were labeled in these regions, such as the BNST, the lateral hypothalamus, the nucleus accumbens, et cetera, all are directly projecting to the ventral midbrain and presumably regulating the activity of dopamine neurons. So what we've been really interested in over the last few years is trying to identify the specific circuits within these upstream regions that are maybe critical for regulating reward and aversion-related phenotypes in animals via their activity or impact on the ventral tegmental area dopamine system. Okay, so how do we go about doing these experiments? All of the work that I'll tell you about today has relied upon really transformative advances in system neuroscience that have occurred over the last decade, using light-gated channels and pumps to selectively modulate the activity of precise neural circuits. So by using optogenetic methods, we can introduce channels and pumps. We can deliver these with viral vectors similar to those that Eric mentioned in his talk. And then we can target these into specified cell types within the brain. So using transgenic mice, we can even take this further and target these with exquisite precision to genetically and molecularly defined cell types. Then by implanting small optical fibers above these regions, we can deliver light via lasers and LEDs that can directly activate or inactivate the cell type that we're interested in. So when I started working with these tools, I had the opportunity to collaborate with the lab of Carl Diceroth, and a number of papers were published at this time in the sort of late 2000s, really demonstrating that endogenous control of the dopamine system in mice and rats can also produce profound reward-seeking behavior. So here, what you can see is that if we put channel rhodopsin into dopamine neurons and apply light to the tissue in vivo, we can measure a very robust increase in dopamine release in the nucleus accumbens, measured with fast-scan cyclic voltammetry. These same stimulation parameters that produce robust release of dopamine also produce a conditioned place preference in vivo in awake and behaving animals. So this was some of the first evidence that endogenous modulation of these dopamine cells can actually produce a conditioned reward-related behavioral phenotype with just endogenous select stimulation of the dopamine system. Additionally, work by Alana Witten and Liz Steinberg that was done a few years later showed that in rats that you could train rats to essentially self-stimulate or lever press to receive optical stimulation directly at the level of the VTA dopamine neurons. And so this is really important because it highlights that endogenous modulation of these cells, not just pharmacological or lesioning approaches, but really modulating the cells that exist within this particular brain region can produce reward-related behavioral phenotypes. So this is critical, but at the same time, we know that the ventral tegmental area, the region of the brain where these dopamine cells reside, is highly heterogeneous, and in fact, only contains about 60% dopamine neurons, and the rest are made up mostly of GABAergic and glutamatergic neurons. And so one of the first projects that we did when I started my lab at UNC Chapel Hill was to start to consider some of these other cell types within the ventral tegmental area because we felt they were less well understood. So we wanted to know what happens when we modulate these GABAergic neurons in the VTA. What we did is to develop a way to target these cells using those same tools. We targeted the GABAergic neurons by using a VGAT-CRE mouse line, which allows us to selectively target these neurons. You can see we're targeting the cells here with green fluorescent protein and staining the same tissue with, against tyrosine hydroxylase, the marker of the dopamine neurons. And you can see that these GABAergic neurons are completely intermixed with the dopamine cells, but essentially non-overlapping. And so what we wanted to do was to start to dissect what happens when we modulate the activity of these neurons. What we did was to train animals on a reward-seeking task that we developed. It's a Pavlovian conditioning task where a tone house light stimulus predicts the delivery of a small sucrose reward that the animals will consume. And what we did is to then modulate the activity of these VTA GABA neurons at critical time points in the task, either when this cue's on or when the animal is actually consuming the reward. What we saw was that in well-trained animals, they learn this task very quickly and learn that when the tone comes on here, that they approach the reward and will consume it. So these raster plots are showing the licks that an individual animal is making, and this histogram below is showing the average data across the session. And what you can see is that when we apply brief optogenetic stimulation of these GABAergic neurons in the VTA, you can see a transient but highly reproducible disruption in their reward-seeking behavior. So this was some of the early work that really was beginning to suggest that these GABAergic neurons have a very different and functional phenotype from the dopamine cells that are in that region. At the same time that our paper was published, a paper from Kelly Tan, who was in Christian Lucher's lab at the time, also demonstrated that optogenetic modulation of these VTA GABA neurons produced a conditioned place aversion when they're stimulated. And moreover, if you inhibit the activity of the dopamine neurons in this same paradigm, you can also see a conditioned place aversion. So this suggested that perhaps these cells might be directly impinging upon and regulating the activity of dopamine neurons, and we wanted to investigate that in more detail. So to do this, we put channel rhodopsin into the GABAergic neurons while performing brain slice electrophysiological recordings from the neighboring dopamine neurons. So if we inject current to activate the dopamine neuron, we can get it to fire a train of action potentials seen here. But if we apply this same current injection in the presence of light that will be activating the VTA GABAergic neurons, you can see we can reduce the activity of the dopamine cells. So this is suggesting that one of the main principal functions of the VTA GABAergic neuron is to act as an inhibitory brake on the dopamine neurons themselves. So next, what we were interested in doing is turning our attention to some of the more distal inputs that are coming into the VTA. And one that we first started working on was projections from a brain region called the lateral habenula. For those of you who don't know, the lateral and medial habenula make up part of the epithalamus. They sort of sit under the hippocampus in the primate mouse and primate and mouse brain. And the lateral habenula in particular sends glutamatergic projections to the two major monoaminergic centers in the brain, mainly the ventral tegmental area, the dopamine system, but also to the dorsal raphe, to the serotonergic system. And so because of this work, and as well as work that was done in non-human primates from the Hikosaka lab, we were interested in testing the functional significance of this lateral habenula to VTA pathway. Now, work from the Hikosaka group, where they recorded from habenula neurons, lateral habenula neurons in non-human primates, suggested that these cells in the lateral habenula are coding for aversive stimuli exposure. So if you present a monkey with an air puff, you can see a transient but significant increase in the activity of these lateral habenula neurons. Now, what's quite interesting is that in another paper, they showed that these lateral habenula neurons almost show an opposite firing pattern to the midbrain dopamine neurons. So dopamine neurons, as probably most of you know, fire initially to unpredicted rewards, but also to cues that predict rewards, whereas habenula neurons are inhibited when rewards are presented. And so this suggests, because they're projecting to the VTA, or some of them are projecting to the VTA, we thought that it's likely that they're indirectly regulating the activity of the dopamine cells through modulating GABAergic neurons. And so we set out to use optogenetic methods to target these cells. So we put chanorodopsin selectively into the lateral habenula. We can evoke very robust firing in these neurons in brain slices. And if you cut a parasagittal slice through the brain, you can see very nice labeling of the axons from the lateral habenula that are coming in to the VTA. And so to test the behavioral relevance of this pathway, what we did was to put chanorodopsin into these areas and then implant an optical fiber directly above the VTA. And so what this allows us to do is to selectively stimulate those lateral habenula fibers that are coming into the VTA without presumably activating other lateral habenula fibers that might be going elsewhere. And so one of the behavioral tests that we've used extensively to assess whether a particular neural circuit may have rewarding or aversive qualities is this real-time place preference assay. It's a very simple behavioral task where an animal is allowed to freely explore two sides of a chamber. This is a control animal that's only expressing yellow fluorescent protein. What you can see is that when the animal enters this side of the chamber, the laser turns on to deliver light to the brain region of interest, whereas on this side of the box, no light is delivered. And what you can see is that in these control animals that are not expressing chanorodopsin, they explore both sides equally. Now, in the chanorodopsin expressing animals, we saw a very different behavioral phenotype. You can see that when the animal enters the side of the chamber that's paired with optical stimulation, they immediately turn around and avoid that side of the box. So this was some of the first evidence to us that this pathway from the lateral habenula to the VTA was likely negatively regulating the activity of dopamine neurons and causing aversion-like behavioral phenotypes. This is just the summary data here. Again, the control animals show very robust or show equal time spent on both sides of the chamber, whereas the chanorodopsin expressing animals only spend a very small amount on the side paired with stimulation. This also evoked escape behavior, where the animals would just try to escape from the side of the box where they received light stimulation. The next experiment we wanted to do was to look at whether or not animals would actually engage in a task where they had to work to alleviate stimulation of this pathway. So you can think of this as a negative reinforcement task. We asked whether or not we could teach these mice to nose poke to essentially turn off a stimulation. And that's essentially what we saw, was that if we had stimulation of the lateral habenula to VTA pathway on, animals would rapidly learn to nose poke to be able to turn this stimulation off. So we think this can serve as a negative reinforcer as well. And then finally, the last experiment from this study that I'll share with you is looking at whether this modulation of this pathway can reduce responding for a positive reinforcer. So in this task, what we did was to train mice to nose poke to receive a small sucrose reward. So you can see the animal nose poking over here and then going and immediately retrieving the reward. Again, this is the control animal. And at the time of the nose poke for the stimulation animals, they received brief stimulation of the lateral habenula to VTA pathway. And so, let's see, it should switch in a second. But essentially what we saw was that when we stimulated this pathway, and you'll see it here now, we saw a very different behavioral phenotype. Instead of immediately retrieving the reward like the control animals do, the animals that receive stimulation essentially back away and don't immediately retrieve the reward. And this is just the summary data of that video. Okay, so up until this point, what I've shown you is that while the dopamine cells are critical for driving motivated behavioral states and reward-seeking behavior, there's at least a few ways which they can be modulated by upstream circuit elements. In particular, the VTA Gabaergic neurons, which are local, as well as the more distal glutamatergic neurons from the lateral habenula, which are directly projecting to and mostly targeting the Gabaergic neurons. So we think that these cells are exerting their aversive properties via driving the activity of the Gabaergic neurons in the VTA. So what I'll do is to tell you about two other additional circuits that we've identified, both of which also interface within this larger brain reward circuit. And so next what we did was to focus on another region of the brain that's been critical for driving different types of behaviors, the bed nucleus of the stria terminalis, which is a part of the extended amygdala. Now, the BNST is probably most well studied and most well understood as a brain region that's critical for controlling anxiety-related behaviors, as well as long-term fear-associated behaviors. At the same time, while it's been classically involved in sort of the aversive behavioral states, it's also known to send very dense projections to the ventral tegmental area. And so we wanted to ask the question whether this BNST to VTA circuit would have behavioral consequences. So what we did in this study was to develop methodology that allowed us to record from BNST neurons in vivo in awake and behaving mice and identify those that are projecting to the ventral tegmental area. So these are now a heat map showing the activity of BNST to VTA projection neurons selectively. And what we did, because of the role of the BNST in processing aversive stimuli, we asked what happened to these cells when we exposed the animal to an unpredictable foot shock stressor. So what you can see is that in this heat plot showing the activity of all the neurons before the shock, during the shock exposure, and after, you can see that we see a very heterogeneous response pattern across all these neurons. Some cells show increases in their activity during this shock period that persists after the shock period is over, while there's other cells that show profound inhibitions in their activity during this. So this suggests that this brain region, like many areas in the brain, is very heterogeneous in its function. Now, we did have some idea about the heterogeneity of this brain region because using tools such as the Allen Brain Atlas, we can identify multiple molecular markers that we think are marking different cell populations. And so in this first study, what we did was to look at two broad classes of neurons that exist within the BNST that also project to the VTA, namely glutamatergic neurons that are marked by the expression of VGLUT2, and GABAergic neurons that are marked by the expression of VGAT. You can see that within the BNST, both of these cells exist. So it's a mixture of both glutamatergic and GABAergic neurons, both of which project to the VTA. Now, what was interesting was that when we recorded selectively from either these glutamatergic neurons that project to the VTA or the GABAergic neurons, we saw that heterogeneity in the responses of the cells was reduced. Namely, the glutamatergic neurons that project to the VTA, most of them showed an increase in their activity in response to shock exposure, while the GABAergic neurons that project to the VTA show predominantly this inhibition phenotype. So based on the neural dynamics of these two different cell types, we hypothesized that they're likely doing very different things. Just also quickly to mention that when we performed patch clamp recordings in the VTA and activated these different populations, these different inputs, we saw again that most of them targeted the non-dopaminergic cells. So we saw that most of the cells that were receiving input from the BNST were actually the VTA GABAergic neurons. So then what we did was to turn our attention to the functional consequences of manipulating these cells. And so again, using the real-time place preference assay, which I've already described, we photostimulated the fibers that are coming from the glutamatergic cells in the BNST to the VTA. Now remember, these were the cells that showed an increase their activity in response to foot shock. And what we saw was that when we artificially stimulated these cells with optogenetics, we saw a very robust real-time place aversion, meaning they would avoid the location paired with the stimulation. In addition, we tested the animals in an open field test, which can be a measure of anxiety-like behavior in mice. And what we saw was that when we activated these glutamatergic neurons from the BNST to the VTA, the animals showed a reduction in the amount of time they spent in the center of the chamber and an increase in the amount of time they spent in the corners of the chamber, which is indicative of an anxiety-like state in mice. So what about the GABAergic neurons within this structure? So again, we targeted the GABAergic neurons in the VTA and selectively activated their fibers that innervate, or in the BNST, and selectively activated the fibers that innervate the VTA. What we saw was essentially the opposite behavioral phenotype to what we saw when we activated the glutamatergic neurons. Namely, if we drove the activity of these GABAergic neurons in the BNST to VTA, we saw that this produced a real-time place preference, so the animals spent most of their time on the side of the chamber that was paired with optical stimulation. And then in this study, the last experiment we wanted to do was, because in vivo, we saw that these GABAergic neurons were showing a reduction in their activity in response to foot shock. We wanted to see if we could sort of buffer some of the fear and anxiety-like behavioral phenotypes by artificially driving the activity of these cells. So what we did to look at this was to stimulate these GABAergic neurons at a time when they're normally showing a suppression in their activity. So when the animals are shocked, these cells are normally showing a reduction in their activity. In this case, we artificially stimulated them at 20 hertz. And then after this shock period, we looked at their post-shock freezing behavior, as well as their behavior in an elevated plus maze. What we saw was that when we stimulated these cells during shock exposure, we could reduce the amount of time that the animal spent frozen after the shock, and they showed an increase in terms of the open arm entries in the elevated plus maze. Both of these behavioral experiments suggest that artificially modulating the activity of these BNST to VTA GABAergic neurons can buffer against the aversive behavioral features following aversive stimulus exposure. Okay, so to just summarize this study, what I showed you is that the activity of the BNST to VTA glutamate cells are enhanced following foot shock, while the activity of the BNST to VTA GABAergic neurons show the opposite effect. They show a reduction in their activity. We think that these are functionally distinct but essentially parallel pathways that are largely or preferentially innervating the non-dopamine cells within the VTA, namely the VTA GABAergic neurons. And then finally, I just showed you that the behavioral consequences of activating these cells is quite different, meaning that when we stimulate the glutamate cells, it produces aversive-like behavioral phenotypes. When we stimulate the GABAergic cells, we see reward and anxiolytic-like behavioral phenotypes. Okay, so the last circuit that I'd like to talk about is one that we've been working on a little bit more recently. And this is really focusing on, again, a population of neurons that are subcortically located that express the gene VGLUT2. So again, these are glutamatergic neurons. In this case, we've been focusing on the lateral hypothalamus. So the lateral hypothalamus has been known for many decades to be a critical neural substrate for driving reward-seeking behavior. And in a few studies over the last couple years, we've been focusing on trying to understand the molecular basis for this by picking out different cell types that exist within this structure, studying who they're connected to, and essentially what they're encoding. So we think that this fits into the sort of canonical brain reward circuitry via the following way. So when we targeted these glutamatergic neurons within the lateral hypothalamus, what we saw was that if we look at where their axons are actually projecting to, you can see that there's very robust innervation to the lateral habenula, which I just talked about. So this right off the bat kind of suggested that this is an excitatory projection that's sending input to the lateral habenula. I just showed you that the lateral habenula is highly aversive, so we predicted that modulating this excitatory input to the lateral habenula would produce aversion-associated behavioral phenotypes. And that's essentially what we saw. You can see, again, in the real-time place preference assay, the animals avoid the side of the box paired with stimulation. If we actually put in an inhibitory opsin, halorhodopsin, which will suppress the activity of these neurons and their projections, you can see that when we suppress the activity of the fibers in the lateral habenula, the animals show a place preference to this. And this is just quantified here. We also looked at whether inhibition of this pathway from the lateral hypothalamus to the lateral habenula altered reward consumption behavioral phenotypes. So typically mice will readily consume highly palatable liquid rewards. And this can be used as a metric for reward seeking as well as anhedonia-like behavior in mice. And so what we saw was that when we blunted the activity of this pathway using inhibitory optogenetics, we saw that this increased the amount of sucrose that the animals would consume within this task. So they would lick more for the sucrose and they would show a higher number of drinking bouts within this task. More recently, Mark Rossi and Marcus Basiri, who are currently in my lab, have been following up on this to investigate what do these cells actually code for, these glutamatergic neurons in the lateral hypothalamus. So to do this, we've been using and developing approaches to use deep brain calcium imaging as a way to assess activity in neurons in a genetically defined way. What's done is that we put the genetically encoded calcium indicator, GCAMP6, into a subset of cells that we're interested in this case, the lateral hypothalamic glutamate neurons. And then we implant a small microendoscopic lens, a GRIN lens, directly above these cells. So this serves as a way to provide optical access to these cells that are located deep within the brain. We can couple this GRIN lens to a 2-photon microscope, which allows us to visualize the activity of these cells. So when the cells show an increase in their fluorescence, this is due to calcium coming into the cell, which typically happens during a burst of action potential. So the general idea is that the fluorescent changes serve as a proxy for the activity within a genetically defined cell type. You can see that using this approach, we can get pretty nice cellular resolution where we can now image the activity of individual neurons in an awake and behaving animal and be able to do so in a genetically precise way. So what we saw was that when we imaged the activity of these cells, these glutamate cells, many of which project to the lateral habenula, we saw that to the receipt of reward, when the animals were actually engaging in the consumatory behavior, we saw that there was an increase in the activity of these cells following time to reward consumption. Now, what was interesting is that the motivated state of the animal seemed to change the activity dynamics of the cell. So one of the powers of this approach is that we can record from essentially the same group of cells or the same cells longitudinally over many days or different sessions. And so you can see that when the animals were ad lib fed and fully sated, we saw an increase in their activity. But when they were hungry and just acutely food restricted, you can see that the activity within these cells was reduced. And so this was really interesting to us because it suggested that when the animals are hungry and more motivated to obtain sucrose, we actually see a reduction in the activity of these cells. Now, the final experiment that Marcus did in my lab was to transiently, optogenetically stimulate these cells just time locked to when the animal was engaged in the consummatory behavior. So only brief optogenetic stimulation was applied at a variety of different frequencies, time locked to when the animal was consuming sucrose. And what you can see is that optogenetic activation, time locked to when they're consuming the reward, leads to a very powerful disruption in their reward-seeking behavior. Across all these frequencies. So at the higher frequency, there's a very severe disruption in the microstructure of the animal's licking. At the lower frequencies, you can see that they lick time lock to the reward and retrieve it. And as we scale up, you can see that this behavior is more and more disrupted. And so what we think is going on within this circuit is that it's via its projections to the lateral habenula, it's actually serving as a brake on consummatory behavior. Okay, so just to summarize what I've told you about today, sort of the big picture take-home points, we think that a variety of genetically defined cell types are indirectly regulating the activity of VTA dopamine neurons that control reward and aversion. Note that most of these projections that I've talked about today aren't directly contacting the dopamine cells themselves, but are all going through the intermediary circuit node of these VTA Gabaergic neurons, we think. Another interesting point is that many of these brain regions, such as the BNST, as well as the lateral hypothalamus, seem to contain excitatory and inhibitory neurons that have opposing behavioral features associated with stimulating these cells. So I mentioned our work in the BNST, but there's also emerging evidence from other subcortical areas from other groups that there's also excitatory inhibitory neurons in the ventral pallidum, as well as those in the LH, in the lateral hypothalamus, that all seem to be having similar features, meaning the GABAergic neurons are associated with reward phenotypes, whereas the glutamatergic neurons are associated with aversive behavioral phenotypes. And so we think that these approaches have identified multiple important functionally relevant circuit components, and now we know what cells we think are critical for driving this, and we think that this could potentially be used as a way to prioritize certain cell types and connections for future translational opportunities. And just to end, I'll just briefly mention one thing that we've been working on as a way to sort of bridge the functional neurocircuitry work with identifying novel therapeutic targets in a cell type specific fashion. So what we've been using is single cell transcriptional profiling, which is a method where we can perform RNA sequencing on thousands and thousands of individual cells that are captured from a single brain region. So typically we would punch out a small area of the brain. We can partition individual cells into nanoliter-sized droplets that are that also contain an RNA capture bead. and then perform standard high-throughput sequencing to isolate the transcriptomes from many thousands of cells that are taken from a single animal. Using principal component analysis and visualization approaches, we can identify within a single brain region a variety of molecularly defined cell types that exist within this structure. So not only do we get different types of neurons, but we also see many supporting cell types, astrocytes, et cetera. And we can leverage this information to identify novel genes that are changed in a cell type specific fashion. And we think that this is going to be a very powerful approach for bridging our work in animal models to identifying novel circuit specific therapeutic targets that might have translational relevance. And so finally, I'd just like to acknowledge everybody in my lab who helped with this work. Alice Damatakis and Josh Jennings were two former graduate students of mine who were largely responsible for the early work I presented. And then I mentioned Marcus Basiri and Mark Rossi, who are working on the unpublished work. And I'd also like to acknowledge our sources of funding. Thank you.
We have time for maybe one question, if anybody has a burning question.
Thank you for this great talk. I've got a question regarding the hypothalamic two-photon work. Maybe I missed this detail. Did you express the calcium indicator only in neurons projecting to the habenula or in all neurons?
Yeah, that's a great question. In the data that I presented, it was expressed in all the glutamatergic neurons in the lateral hypothalamus. We're doing follow-up studies now to do the projection-specific targeting.
Thank you.
Yeah.